Okay, sir, I think we can get started now.
Hello. Yep, I'm ready.
Okay, so welcome everyone to the fifth Astrobio.
Arjun, we have
Yeah.
to start streaming.
Oh, okay, fine. Yeah. Tell me once.
Just a second, yeah.
Let's just start. Yeah.
So is it okay if we record this talk?
Yes, that's no problem.
Sharang, has the stream started?
Okay, so welcome everyone to the fifth astrobiology talk hosted jointly by the Ecological Society of IISER-TVM and Parsec, the Astronomy Club. We also thank Anvesha, the Science Club, and the Physics Department of IISER-TVM for helping us in achieving this mini series. Today we have Dr. Ivan Glaucio Paulino Lima, who is a research scientist at Blue Marble Institute of Science and NASA Ames Research Center. His research topic is related to extremophiles and their adaptations to extreme environments. He is building a database of reflectance spectra that will help astronomers to look for biosignatures and exoplanets. He's also working on the Powercell experiment on both the EuCROPIS satellite, which is currently testing biological experiments in Earth's orbit under 3 different gravity regimes. His role in the mission involves the testing of bioengineered bacterium Bacillus subtilis for flight and developing a robust and automated genetic transformation protocol. We are very thrilled to have you aboard, Dr. Ivan Lima. You could carry on with the talk.
All right, everyone, thank you very much. I would like to thank everyone for this invitation, for this opportunity to talk a little bit about my work. And since we don't have a time constraint really for this event, feel free to interrupt me if you have questions. So I prepared a presentation. If I go through the presentation without questions, it will take like 1 hour. But feel free to ask me questions or save questions for later if we have time. So I'm going to start sharing my screen right now. And please let me know if you can see it.
Yes, it's visible.
Yeah, okay. So this is the title of my presentation, Extremophiles and Space Exploration. So I'm gonna give a brief overview about what extremophiles are and how they relate to space exploration. So I belong to this institute, Blue Marble Space Institute of Science, which is a research institute formed by enthusiastic researchers, and they are all working in very exciting areas related to astrobiology and space exploration. Many of them I knew before joining the institute, and we recently had Carl Pucher, the former the head of Astrobiology Institute at NASA, to join our institute. So it's a very exciting work environment to be able to interact with all these people. and know more about their work. Extremophiles are living beings that are adapted to live and thrive in limits where the physical chemical parameters are very extreme. And some people think that this is like an anthropocentric concept, because what are extreme for us may not be extreme for these living beings. But if you think about the limits of stability of molecules, the physical chemical limits, they live very close to what is even possible for these type of molecules, like organic molecules. Some of these conditions are very, very harsh. If you think about the deep ocean, where pressures are very, very high and temperatures as well, and also all sorts of environments that we can go, like the Atacama Desert, very, very dry environment, the Bolivian Altiplanos, the Antarctica, and the hot springs. Whatever we can find water, we will find some form of life thriving in these very harsh environments. So that's why NASA used this follow the water mantra to look for signs of life beyond Earth. So extremophiles are not an anthropocentric concept. They live in very close to what is physically possible for the stability of molecules. And I had the opportunity to participate in field expeditions for some of these environments. This one is Mono Lake in California, where the water is very high pH, so it's very alkaline, and we can still find many forms of life thriving there. And here is another example, iron mine in Colorado, where we can find a very acidic environment. And the objective here was to isolate phototrophs, photosynthetic microorganisms that are able to thrive in this very acidic environment. And another example, the Atacama Desert in Chile, where I collected some samples for my own work as a postdoc at NASA. Very, very dry environment, as you can see, no vegetation, and very, very hot and dry. It's even difficult to breathe there if you are there for more than 2 hours. So I have been working at NASA since 2011. I joined Dr. Lynn Rothschild's lab as a postdoc. And as you can see here, her lab is a very diverse lab with different lines of research, from biowires and biomining to biomaterials and bioprinting. We have this work on synthetic biology and its application beyond Earth, and human exploration, mostly in situ resource utilization using cyanobacteria, cell-free systems, origin of life, halophiles, and limits of life. This is particularly interesting to me. This is the area of research where I work most, looking into extremophiles and especially radiation. Miniature electrochromatographic and monolithic columns. So all this miniaturization and automation of processes that we can think of in the laboratory, we try to develop in our lab. Search for life beyond Earth. And here there is this International Genetic Engineered Machines competition, which is held every year since 2011 in our lab, in Lynn Rothschild's lab. And it is basically a student competition in synthetic biology. We didn't have, of course, last year because of the pandemics, and not this year as well. But we had many very interesting projects, like self-folding origami, biodrone, bioballoon, and also EuCROPIS, which led to a spaceflight mission that I'm involved right now. And these students, they come from Stanford University and Brown University. Those are the universities where Dr. Lynn Rothschild is faculty. So she brings students from these institutions to do these research projects in the summer in our lab. So extremophiles are a very prominent area of research within astrobiology. And in 2015, NASA published this document called Astrobiology Strategy, which is basically a survey of areas of research that will receive funding for research within the next 10 years. And it's available on this website. And it brings a very nice table that relates all the different kinds of extremophiles, considering different factors like high temperature and low temperature, high pH and low pH, and so on, and the different classes of extremophiles, their defining growth conditions, and some examples of organisms. Most of them, as you can see here, are microorganisms, but some of them are not necessarily microorganisms. There are other forms of life that are not microorganisms that are considered extremophiles. And one classic example is tardigrades. They are animals that live in very dry conditions. They can survive space conditions, very high radiation and dehydration, and they are able to rehydrate and resume their metabolism. But most of them are microorganisms, like any type of environment. You know, even if we think about our own body, there are more microorganisms living inside our bodies than our own cells. And this is true for any kind of environments, including extreme environments, which actually microorganisms dominate extreme environments. The most extreme environments, the most simple the life form will be. And from all of those different types of organisms, those that are interesting to me are the ones that are radiation resistant, like Deinococcus radiodurans, Rubberbacter, and Purococcus furiosus. And for my PhD in Brazil, I studied the survival limits of Deinococcus radiodurans to extreme radiation conditions like those found in other planets and in the interplanetary medium. So why the study of radiation and its effects on materials and biology is important? We know that radiation from space, from the sun, can affect satellite communications and prevent our ability to communicate through satellites. And this is a very big issue for satellite communications. But of course, as a biologist, the ones that are more interesting to me are the ones that affect biology, right? And their effect, the effects of radiation is the most important ones are on DNA, because they can change genetic information and generate mutations or lead to cell death. And to give some perspective on the radiation environment beyond Earth, there is this very nice chart in Science in 2013 comparing radiation exposure from different sources and different conditions. For example, we can start here at the top. An average citizen in the US is exposed to 3.6 millisieverts in a year, just by being anywhere in any kind of environment. That's the natural background radiation, you know. If you take a CT scan, abdominal CT scan, you are exposed to 8 millisieverts. Department of Energy radiation worker annual limit is 20 millisieverts, and an astronaut during 6 months trip to the ISS is exposed to 75 millisieverts, and this is the radiation measured by the Mars Science Laboratory radiation instrument on transit to Mars during 7 months. So it's 466 millisieverts, which is way beyond any type of exposure we have here on Earth or on ISS. So ISS is within the radiation protection from the magnetic field of Earth. So that's why they don't have such a large amount of radiation. It's still significantly higher than on Earth's surface, but they are still protected within the magnetic field from Earth. But as you can see here, a trip to Mars would require some type of protection or mitigation of the effects of radiation. And it's very expensive to protect against radiation through shielding, because it's heavy. Any type of shielding will be heavy. Like if you think of lead, which is very common in radiation environments, in reactors, radioactive reactors on Earth, it's very, very heavy. Even water, water is very heavy to bring up to space, and you can shield a lot, but some types of radiation will still penetrate any type of shielding. So you really need some kind of strategy to countermeasure radiation. And we know that space has different sources of radiation. We know that the sun emits not only light, visible light, and ultraviolet, but also some small levels of X-rays and more energetic radiation. But we also have galactic cosmic rays, and all these types of very energetic rays. They produce a cascade of secondary particles in the atmosphere, and those can also reach the surface and interact with biology. And some people think that these have played a role on evolution of life on Earth. So that's just to show that we are exposed to the galactic cosmic rays all the time. And this is even more true in space. So the sun, as I said, emits visible light and also ultraviolet radiation. But the Earth's atmosphere, it blocks most of the UV. It only passes UV and a little bit of UVA and a little bit of UVB. We don't find any UVC on Earth's surface. And UVC is a type of radiation that we can find in laboratories, in the germicidal lamp that we use to sterilize surface, because UVC has a maximum peak around 254 nanometers. This is the peak emission of these germicidal lamps. And this is very close to the maximum absorption of DNA of any type of life. DNA absorbs at 260 nanometers. That's why this is very effective in inactivate organisms. So we did an experiment in my PhD to test the limits of Deinococcus radiodurans using a synchrotron light source that could deliver vacuum ultraviolet radiation. You can see here in this chart. There is some kind of radiation that is even more energetic than UVC, which is called vacuum ultraviolet radiation. Why vacuum? Because they can only propagate in vacuum. If you have any type of air or any type of surface, it will be absorbed. So it needs to be delivered under vacuum. And this is the kind of ultraviolet that we can find in space because there's no air in space. So we simulated this environment in the laboratory, the Brazilian synchrotron laboratory, and we did exposures of Deinococcus radiodurans under different conditions, like a monolayer, we can see here on A, and also some level of piling up of cells to investigate whether this condition could protect the cells beneath, and also cells mixed with culture medium to see if the organics present on the medium in the medium could protect these cells. And we found that if you have like any layers of cells, it can already protect against the effects of ultraviolet radiation, because this type of radiation, it stops at the first layer of the cell. It's so energetic. It can exchange energy and be stopped in the first layer. It's very deleterious, but it can be blocked very easily. And this is true as well for all the ultraviolet spectrum. If you have like multi-layers of cells, it's already a condition that is beneficial for the cells beneath. But of course, as I said, the space has other sources of radiation, not only the electromagnetic radiation, but also particle radiation. For example, the solar wind, the radiation that comes from the sun regularly, This is called solar wind, and is composed by 96% of protons and electrons and 4% of alpha particles, with the energy of 3 kilo-electron-volts. That's the speed of the particles, basically. But of course, the sun is a pretty stable star, but it has some variations as well. There's an 11-year cycle where the activity of the sun changes. And during these cycles, it can emit solar flares and solar energetic particle events, and then the energies would increase like 1,000 times. But these events are occasional, and the solar wind is constant, you know. So the objective of our research was to test whether solar wind could be a factor that could inactivate all cells in space. So we mixed cells of Deinococcus radiodurans with dust grains of basalt and sandstone. So basalt is represented in all these charts by B or BAS, and sandstone is SST. And because these are two extremes of the different types of rocks that we can find here on Earth. So we wanted to see if there's any difference between these two types, very different types of dust grains. And NC means naked cells, so it was our control for this experiment. And we see here in the top, there's this chart in the middle. We see the inactivation curve of Deinococcus radiodurans exposed to 200 kilo-electron-volts protons. So this curve, the survival fraction is represented here in an exponential scale, and the dose is also in exponential scale here on the x-axis. And we also tested this forsterite. It's a mineral that is commonly found on meteorites, and this is represented by this small cross here. And we can see that the grains, they can protect the cells against this type of radiation, and the doses that they can protect these cells are equivalent to 100 years in space. So this level of radiation is simulating these solar energetic particles, so very energetic events that are common from the sun. And this is a Monte Carlo simulation that shows that this type of particles can penetrate the whole cells. That's why the cells get inactivated through time as we can increase the doses. But if we analyze the solar wind, the 2 kilo-electron-volt electrons and 4 kilo-electron-volt carbon ions, we see that there's no difference between naked cells and grains. They remain all 100% survivors, even at doses equivalent to 1,000 years in space and 20 years in space for 4 kilo-electron-volt carbon ions. And these are the highest dose tested. It doesn't mean that after that they will get killed. And why is that? Because the radiation is stopped at the first layer of the cell, in the cell wall, and they are not able to penetrate. So the cells can easily repair this when they are rehydrated and be able to grow again. So this shows that the radiation present in space are not necessarily enough to inactivate cells, which makes things like the lithopanspermia hypothesis something plausible. We don't know if it really happened in the past, but it is possible that an event like an asteroid impact in one planet could eject materials containing microorganisms. These microorganisms could stay in space for long periods of time, like many years, and these fragments could reach another type of environment, another planet, and if the environments are conceivable for life, they will likely grow and start a life history in this other environment. And here is a schematics that shows how radiation interacts with the cells and what the effects of radiation, the general effects, right? So when we think about ionizing radiation, which are radiation with energy enough to ionize molecules, so high energy radiation, they can have direct effects and indirect effects on the cells. So direct effects are the effects caused on the DNA, which can lead to DNA damage. And so the DNA lose the ability to repair itself. It can generate a change of genetic information, which can lead to mutation or cell death. And the indirect effects are the effects of radiation in other molecules like water that produce a lot of free radicals like hydroxyl, protons, and nitric oxide radicals, and superoxide and hydroxy peroxide, and so on. All of these can have effects on enzymes and membrane, which could lead to DNA damage and also mutation, and also ultimately cell death. So these are the two types of radiation effects on cells, and these effects are influenced by environmental parameters like concentration of oxygen, the temperature, and pH. So it is also affected by the type of environment that these cells are existing. And here is basically the same concept, but this chart is comparing resistant cells in the bottom and a sensitive cell on the top. So here is the radiation coming from the left, generating a lot of free radicals. The hydrogen peroxide and superoxide are radicals that have more site-specific protein damage. So these proteins lose the ability to repair the DNA, and leading to DNA repair malfunction and cell death. In the case of resistant cells, the cells accumulate antioxidant metabolites complexes of manganese. So they are able to accumulate manganese and these organic compounds in the cells. And these compounds protect the enzymes, the DNA repair machinery, and they can repair their cellular functions. They repair the DNA damage caused by radiation and restore the genome and continue with their life cycles. And to give an example, Deinococcus radiodurans can have its DNA shattered by thousands of fragments, and they can repair these damages within 4 hours. They accumulate manganese and have these manganese complexes that protect their protein, but not their DNA. The DNA is shattered, and they are able to restore genomic information, and be able to thrive, even with very, very high doses of radiation. And these are schematics of these manganese complex. What are they? So manganese ions, they tend to combine with nucleoside analogs and orthophosphate and amino acids and peptides, and form these types of things, of compounds. And these act as scavengers of the effects of free radicals, and minimizing the effects of the oxidative stress after radiation. And the radiation effects on cells are very similar to the dehydration effects. If you think about a cell that goes through dehydration, its conformation, cellular conformation is altered, so it loses a lot of water. So the reactive oxygen species inside the cells accumulate, and the cell undergoes biochemical stresses, metabolic stresses, physical stresses, and physiological stresses. And one of the main effects are on DNA. They can have double-strand breaks and single-strand breaks, and which is the same type of damage that is caused by radiation. So for my postdoc, we thought about this question: is Deinococcus radiodurans the last frontier of radiation resistance, or other organisms could be thriving out there and hiding some other strategies to survive radiation? So other organisms could accumulate higher levels of manganese. They may have a more efficient DNA repair toolset, and may produce unusual oxidant secondary metabolites. So I went to different types of environments. This is the picture that I already showed you, to the Atacama Desert, and I also went to the Sonoran Desert, where we had a film crew from NHK, from Japan, and there's one episode for Mars terraformation. I think it's episode number 18 of the first season of Cosmic Front. There is a little bit of information, more information about this research there. So basically, I collected samples from these environments and exposed to the germicidal lamp in the lab, UVC exposure, to 300 joules per square meter, which is very, very high. It's like 10 times higher than what a regular microorganism could withstand in the lab. And we use this dose to select organisms that are resistant to radiation. And we found many, many organisms after cultivating them in different types of culture medium in agar plates. And this chart on the left, we show that different types of environments, they yield different variety of organisms. So we have here in the x-axis, here in the bottom, different types of samples, the Ames Research Campus samples, manganese mine in Sonoran Desert, and the Atacama Desert here. And we also have this F and S, which are filtration and sprinkling methods, two different methods that we used for these environments. And we see here that clearly the Atacama Desert is the sample that showed the highest microbial diversity of organisms. And we show this diversity here by the representative genus that we found in the Atacama Desert, and which is higher compared to the manganese mine in Arizona and to the Ames Research Campus at NASA in Northern California. So then we ranked all these organisms by their ability to withstand another round of 300 joules per square meter, this time with a controlled experiment with isolated organisms in a liquid solution, so very controlled experiment. And so we ranked all these organisms, and we see that Deinococcus is still like the champion here, withstand very nicely, like 100% survivability to 300 joules per square meter, and we have this gradient of resistance to this level of radiation. And we see here all this microbial diversity of organisms, and most of them are pigmented. They live in a desert, exposed to high UV, so they have to have some sort of strategy to protect them against the effects of radiation. And then we compared the extracellular and intracellular manganese to iron ratio versus the survival of each one of these organisms individually. And we found that the concentration of manganese or iron in the environment are not related to the survival capacity of these organisms, but the intracellular manganese to iron ratio is related to the capacity, to the survivability of these organisms to UVC radiation. And why iron? I didn't mention that previously, but iron generates a lot of phantom reaction in the cell, which generates a lot of oxidative stress. So if an organism has a lot of manganese in the cell, but also has a lot of iron, the manganese to iron ratio will not be very high, and the organism will not be very resistant to radiation. But if the concentration of iron is low and manganese is high, then the organism tends to be very resistant to radiation. This was already demonstrated by laboratory strains for ionizing radiation, and our study was the first one to demonstrate that this is also true for environment isolates and for other type of radiation, which is UVC radiation. And so two of these organisms, Hymenobacter and Geldermatophilus, were the highest, the most radiation resistant organisms in our collection. and we compared them to Deinococcus radiodurans, we see here that Hymenobacter showed in red is even more resistant than Deinococcus radiodurans to UVC radiation. And the Geodermatophilus is also more resistant in the highest doses. And we will see why in a minute. Deinococcus still remain the champion for ionizing radiation, so they are very, very resistant, but still Hymenobacter and Geodermatophilus are two organisms that resist doses like thousands of times higher than regular organisms like human cells in vitro. So Hymenobacter, it doesn't produce a lot of pigments. They produce a very light pink pigment. So we think that the main mechanism that contributes for its high survival against UVC radiation is DNA repair. So that they may have some very, very efficient DNA machinery to cope with high doses of radiation. And here on the right, Geodermatophilus obscurus, or Geodermatophilus, our other organism, it forms cell aggregates, and they have this very, very dark pigment. So they can be protected passively against radiation. That's their strategy to survive these high doses. And these organisms are being currently studied at the molecular level, at the genomic level, in a partnership with people from our collaborators in Japan. So this study was published in the Journal of Photochemistry and Photobiology B in 2016. And I'm happy to share this manuscript with all of you. So this is our collaborator in Japan, Kazuharu Arakawa from the Institute for Advanced Biosciences in Keio University. And what we are doing is conducting a genomic comparison between organisms from two genera, Cocuria and Astrobacter. So we repeated the experiment for these isolates, and we isolated the genomic, we did whole genomic sequencing for all these organisms. And we are doing a comparative genomic study to see exactly what kind of genomic difference they have to show these different radiation resistant profiles. One of them is already published in the Journal of Genomics, and more will come in the next months and years. So what I'm very much interested now is the not yet cultivated microorganisms. So I'm sure that there could be organisms more resistant than Deinococcus radiodurans. If we think about the unculturable bacteria, which is not necessarily unculturable, we actually fail to cultivate them, but efforts for cultivating the highest diversity of organisms are being conducted throughout the world, designing culture medium more efficient to retrieve these organisms. And one question that we have is how to test for radiation resistance in organisms that we are not able to cultivate. If you think in a natural environment, there could be like a network of relationship between different types of organisms where a radiation-resistant organism could need a compound that is produced by a radiation-sensitive bacteria. And if you irradiate an environmental sample, you will lose a radiation-resistant organism, not because it died because of radiation, but because it doesn't have a compound that is produced by a radiation-sensitive organism. So everything we know right now about radiation resistance is by the knowledge that we can build in the lab, cultivating microorganisms and doing these survival curves. And one thing that we came up is to use DNA dye, a dye that form complexes with DNA, and it can only penetrate dead cells. It cannot penetrate live cells. So if you take an environmental sample and you irradiate this sample and then treat with this dye, which is called PMA, propidium monoazide, you will only amplify the genetic information that are from the organism that did not suffer the effects of radiation. So you only retrieve information from the ones that are resistant to radiation. And this is a project idea that we are developing right now and applying for grants, for funding. And I think this could lead could dramatically improve our the amount of information that we have from at the genomic level from samples in the different types of environments. And we recently published a review of tools for distinguishing the living from the dead in microbial ecosystems, where we have more information on this dye in this manuscript. As I mentioned, the laboratory where I work there at NASA Ames is heavily involved in synthetic biology. It's actually the synthetic biology lab at NASA. We use different techniques to produce for different applications related to space exploration. And NASA recognizes that the potential of synthetic biology for space exploration. And this is a very nice chart retrieved from this website here at the bottom that shows like a concept of what could be astronaut outpost on Mars, for example. We know that the cyanobacteria, represented here in this green greenhouse here, they are very cheap to cultivate because they only need light and some micronutrients and minerals to be able to grow, and they can produce a lot of different things, and we can improve this process with synthetic biology. But the most dramatic example of the potential of synthetic biology for space exploration is, imagine that an astronaut becomes ill on Mars and require like a biological solution for this illness. There's a limited capability in this colony or in this outpost to produce the biological solution. But if we transmit this information back to Earth, there's a larger network of laboratories that could work on a biological solution, and this solution could be transmitted remotely, and then this information could lead to the development of the biological solution there on Mars without the need to ship anything between the two planets. So that's the power, that's the vision for the synthetic biology in space exploration. So this is what I was talking about, cyanobacteria. It's a very nice biological model to think about projects for space exploration because it requires solar radiation. It can use CO2, and some of them are also diazotrophic, meaning that they can also fix nitrogen. They need a little bit of water and micronutrients and minerals that are present on Mars, and also energy and heat that is produced by solar radiation and can be enhanced by different technologies. And they can produce organics and other materials and compounds that can be used by heterotrophs to produce food and pharmaceuticals and different types of materials that will be needed for space exploration. And based on this concept, the 2011 iGEM team in our laboratory thought about doing an experiment to introduce the capability into a cyanobacterium called Anabena to produce and secrete sucrose, which is a sugar that could be then used by another organism like Bacillus subtilis to produce food, fuel, clothes, drugs, bricks, and etc. So they did that. They genetically engineered this Anabena to produce and secrete sucrose. And then started the experiments in 2011. In 2013, we had a visit from DLR representatives, the German Aerospace agency, talking about an opportunity to fly an experiment in their satellite platform. And our project was selected, and we started to design this experiment for a space mission in this satellite platform. And we basically tested 4 experiments. The first one we called Powercell, which is the same name as the iGEM team project. Powercell because it's one cell for, providing power, nutrient power, if you will, to other organisms to be able to grow. So that's why Powercell. In order to demonstrate the Powercell concept, photosynthetic organism feeding a production cell, Bacillus subtilis in this case. And this paves the way for human exploration of Mars or Moon, or the Moon. And the second experiment was a transformation, genetic transformation, introducing a new genetic information in an organism. So it is the first transformation experiment in space under 3 gravity regimes. And I'm going to talk a little bit more about that in a minute. It paves the way for synthetic biology technology in space. The third experiment is the protein production based on the beta-glucuronidase gene report system. It paves the way for bioproduction of Earth. And our fourth experiment is our positive control, basically growth in LB. Demonstrate biotechnology reliability in space and paves the way for microbiology in space. So everything, all these experiments are being conducted in a microfluidic plate or a microfluidic card, which fits in a hand. So it's basically 48 wells, 4 rows of 12 wells. Each well fits like 70 microliters of culture medium. The samples are loaded dry in each well and are filled with nutrients after they reach space. So this is a view, lateral view of this microfluidic card, showing how the principle works. So we have the organisms loaded dry in each well, and then after the experiment starts, each well is filled with these fluids, and then they start to grow in the right temperature. I think it was 30 degrees Celsius. And then we start immediately, actually, even during fill, we start the measurements every 15 minutes. I think during fill is more frequent. I think it's every minute or something. And then after it's filled, it's every 15 minutes we have a measurement of the amount of light that passes through these wells. We have a detector at the bottom. And then based on the absorbance measurement, through time, we can build growth curves. And then depending on the experiment, we can interpret these results for the different types of experiments. So we have here a general overview of the arrangement of all these different types of different experiments that I mentioned. We have like controllers, all the hardware components was designed and built at NASA. We actually had a little bit of inheritance from a previous flight called PharmaSat, so it's very similar in design. Here's all the general schematics. So we had actually 4 microfluidic cards, 2 in each payload enclosure. Everything was controlled remotely. We had all this automation, all this automated system to control the experiments, and we could initiate the experiments here from Earth. So this microfluidic card was involved in what's called a payload module, which had the thermal heaters and the LED board and the detectors. And these was placed 2 cards in each payload enclosure. It has this semi-cylindric shape because it was placed on the sides of a cylinder, which was the German satellite. And the satellite itself had a sun-pointing direction. So it had a spin axis pointed to the sun to be able to collect solar radiation in their solar panels. So they had this capability to rotate the satellite in this axis to simulate different levels of gravity by centrifugation, by the centrifugal force. So we were able to simulate the gravity of Mars and Moon, and also microgravity without rotation. So here it is, the satellite under construction there a few years ago at the German Aerospace Center with the solar panels. And here is the cylinder and our payload here integrated in the satellite. So we were the secondary payload. We were basically taking a ride with the Germans in their satellite platform. So after we do all these experiments and all these studies, all these optimizations, we have to load everything and integrate everything in the satellite, and then do a vibration test, which we call shake and vibe test. And of course, we are all happy when everything goes well and everything tests. Every test are positive, you know, so everything is good to go. Sometimes we have to go back and replace things, and it's very good reasons to be happy here. Here's the control room in Germany. I think here is in Bremen with the whole mission team, the DLR team for this mission. And here are the different logos of this mission. Here's the German Aerospace Agency logo here in the top left, showing the experiment, which is basically growth of tomatoes and an algae called Euglena. This is our logo at NASA, the Powercell, showing a Petri dish with Cyanobacteria, and the Moon and Mars, which are the two gravity levels that we simulated in flight. Here's the logo from the SSOA company, actually a space flight company. And this is the mission that carried, I think it was 63 satellites from different nations. I think there's one from India here and one from Brazil, but I was working in one from NASA, the secondary payload. And here's the logo from SpaceX, the Space Flight SSOA Small Satellite Express. So the experiments were launched from a Falcon 9 rocket from the Vandenberg launch base in California, Northern California, Central California. So I'm going to show now.
Your voice is blocked out. I think your mic is muted.
That happens with Google Meet, I think. Every time I unplug my earplugs, it happens. So, but I need to do that to show this nice movie that was produced by Blue Marble. And let me know if you don't hear it.
Stage 2 is present. Right. Launch will drop on that one. We'll go for launch. Three, two,
Okay, let me come back to the presentation and make sure you're listening to me. Yes. Okay, let me go back to the presentation. So this movie, this short video was produced by Blue Marble, which was amazing because one of the things that I didn't mention that the potential for other types of applications on Mars, not only producing all these sorts of things that will be needed, but also biomining, like extracting resource from Mars to build constructions, houses and stations. So microorganisms here on Earth are employed in mining companies to do the extraction of minerals. And we believe that synthetic biology has the potential to enhance this capability of microorganisms as well. So here are some mission highlights. We have over 400 days worth of mission data, 50% flight data, and 50% ground control data. We observed microbial growth in all 4 gravity regimes. We have successful genetic transformation in microgravity, successful growth of Bacillus subtilis and on cyanobacteria cell extract in 3 out of 4 gravity regimes, all hardware and software working as planned, demonstrated ability of hardy bacterial spores to recover from long-term stasis period, which will be fundamental for human deep space missions. Valuable long-term reagent stability data, including antibiotic efficacy, and numerous lessons learned from hardware design to experimental operations are leading to improved design and capabilities for future missions. And we were awarded in 2019 for the successful development and spaceflight of the first synthetic biology experiment, Powercell, aboard the EuCROPIS spacecraft. So I'm going towards the end of my presentation, and I would like to mention some very exciting opportunities for young scientists like this audience. Blue Marble holds a program called Young Scientist Program, which is open for people from all over the world. So you can check out this website, www.bmsis.org/YSP. And you will see a list of different projects available to apply. So you can pick one that interests more for you, and you can get in touch with the PI and think about developing a project remotely. And you receive a certificate from Blue Marble, and you have this experience to exchange knowledge with researchers working in space exploration in very exciting topics. So I would like you to check this website and see all the opportunities that are out there. And another opportunity for students, which is more specific to India, is this Mars Amity Research Station. They have an Earth and Space Exploration program, which is a wonderful opportunity on astrobiology. Hands-on experience, field work, field expedition to the Ladakh region. So very high altitude, very, very exciting place. It's considered one of the Mars analog environments. So you'll be able to experience exploration, hands-on experience. And I think the students stay for 1 week during this between June and October. So you can check it out. Go to this website, www.acoea.com/Mars, and see how to apply. I think there's a registration fee, but you can check it out in this webpage. I would like to be a student and be able to apply for a program like this. And I would like to finish my presentation with this thought from Konstantin Tsiolkovsky, one of the fathers of astronautics. He used to say that the Earth is the cradle of humanity, but one cannot live in a cradle forever. And you might think that, okay, so we are gonna just throw away our Earth and go move to another cradle. That's not the idea. There's a false dichotomy between space exploration and preservation of environment. The Earth will always be the best place for humanity because we evolved here. All the conditions are appropriate for human to live here. But this doesn't mean that we will remain here forever. we know that there are other places in the universe, in our solar system, that we may be able to explore and to set settlements in these places and be able to expand our presence in the universe, not simply move into another place, but expand our presence in order to be able to resist the environmental challenges that we know that happened in the past here. So I think once we know that there are different places available out there, it's a matter of time until we develop technology and be able to have the capability to explore and settle these environments. So I would like to thank you again for this opportunity, and I'm available. I'll be happy to answer questions if you have any. Thank you very much.
Thank you for the wonderful talk, sir. Also, thank you for the valuable suggestions at the end for young scientists like ourselves over here. I had a question on the discovery of phosphine gas last year on Venus, but it turned out to be a false alarm later on, right? But what if it was true, and like what kind of experiments would be conducted, and what kind of simulations would be done on the lab, and what kind of organisms would it be tested on? That was my question.
Yeah. Okay. So the case for phosphine on Venus is still an ongoing debate. I think there are publications that confirmed the result and others that confronted the findings. So I think we are still on the level of confirming, really knowing that there are phosphine molecules in the atmosphere of Venus. So the environment, from a biological point of view, is very, very threatening. It's very, very harsh, very tough for any type of organism. So I think experiments that we can do in the lab would be testing different types of organisms that we think could survive and thrive. That specific region, it's a layer in the atmosphere of Venus where the temperature is conducive for, it's appropriate for life forms as we know them. It doesn't mean that other parameters are also present, like water. So it's a very, very dry place. So there's not plenty of water. It's also very acidic, like extremely acidic. There's no environment, natural environment here on Earth that is as acidic as that type of environment on Venus. It rains sulfuric acid. It's really, really, really challenging. So I don't know if the experiments here on Earth would be even comparable to that type of environment. But we can still do like experiments to see what is the limit, what would be the limit of life forms trying to simulate that type of environment. I think this has value, and we can test different types of organisms, the acidophiles, and also organisms that can thrive dehydration, which is called exoplanets, I think xerophiles. Yeah, so there is a lot that we can still test in the lab and make an argument that phosphine molecules could be being produced by life forms on Venus. But I think it's really, really challenging. I think there are other things that we can do to confirm that there are phosphine molecules on Venus.
Okay, thank you, sir. And there's a question in the chat box. It says, what exactly is a psychrophile?
Psychrophile is basically organisms adapted to very, very low temperatures. So cold environments like the Antarctica and the North Pole, and also very high altitude environments. If you think about the highest mountains, they are always frozen. And there's a specific type of environment called permafrost, environments that are permanently frozen. That's why they're called permafrost. So psychrophiles are organisms that inhabit this kind of environments with very cold temperatures.
Sir, hello? Can you hear?
Yeah. Yes,
Sir,
I can.
They exhibit a stasis mechanism of evolution, right?
The what? Sorry, say that again.
That's stasis, right? In the stasis mechanism of evolution.
stasis mechanism of evolution?
Okay. No, that's. I mean, it's frost for the very longer period of time, right? Permafrost, so it will remain as it is, the environment.
Yeah, frozen for long periods. Yeah, that's permafrost. Yeah.
Okay, sir, got it.
Okay.
Hello, sir.
Yeah.
Sir, question. What led you to think about the experiments for using sandstone and basalt and other materials along with microorganisms? Like, what was the first thought which led you to the experiments?
So basically, the experiment for the flight mission was conceived by students from Stanford and Brown. So they came up with this idea to transform an avena with a transporter, sucrose transporter, so and also something to make them produce sucrose and secrete this sucrose to the medium. And then eventually this experiment became a space flight mission. I think they were already thinking about space exploration, but they had no idea that this could really become a space flight mission. But the idea was to be able to demonstrate that we can use synthetic biology in a project that could benefit humanity because it has some potential space exploration applications.
Okay, so I also have a few other questions. Like, can cells enter a state of quiescence when they are sent to space or something? Like,
yeah, can cells have what?
Can cells enter a state of quiescence when they are sent to space, like dormancy?
Dormancy, yeah,
Yes.
yeah. So, for example, Bacillus subtilis, it was already exposed to the space environment, to the outside layer of satellite. It was a satellite for up to 5 years in space. So, of course, the surface layer was inactivated by ultraviolet radiation from the sun, which is the most deleterious environmental parameter in space. But it can be blocked, as I showed, within like micrometers layers of organics or cells. So the cells beneath that was dormant for up to 5 years, and they were recovered and brought back to Earth by, I think it was a German experiment. Gerda Horneck was the PI on this experiment, and they were able to retrieve. So they can withstand a very, very long period of dormancy. We don't know how long because we haven't tested for such long periods. But I think there is a limit if you think about radiation inactivation or inactivation by radiation. When they are dormant, there's no metabolism to repair the damage, so they accumulate damages. So through time, this damage is accumulated, and when they encounter an environment that they can rehydrate and resume metabolism, there will be such a high amount of damage that they won't be able to survive. So there is a theoretical limit, but we didn't conduce experiments for such long times. But they are very, very resistant in dormancy for like decades, for sure.
Sir, a follow-up to the same question. Like, if we send some signal molecules inside the cell, which might be activated if the cells get out of the dormancy. Like, so can we detect for some certain atmospheric conditions on a particular patch of land on Mars or something? Yes,
we can do such kind of experiment to like a promoter that is inducible by germination. So when they start to germinate, meaning coming out of dormancy, they produce a signal like a color or some sort of signal, and these could be an indication of metabolism or metabolic activity. We can think about such kind of experiments. Yeah, totally plausible.
Sir, I actually lost my connection when you started the explanation. Can you please repeat?
Yeah, sure, no problem. I think it's a good idea to introduce some sort of signal, like a promoter, a germination promoter, promoter that induces germination, and then couple that with a signal molecule that produces light or something, some sort of signal, and this would be an indication of metabolic activity, like cells getting out of dormancy and starting their metabolism, and this could be used for example, in simulation experiments regarding the case for phosphine in Venus. Yes.
Sir, I also have a few questions from my friends who couldn't switch on their mic due to some technical issue.
Okay.
How do different gravitation environments affect the survival of extremophiles?
How to define gravitation environments?
No, how do different gravitational environments affect the survival of extremophiles?
Sorry, what is the effect of gravity?
Yes, what is the effect of gravity, like different gravitational environments on the survival of extremophiles?
Yes, so that's one of the things that we try to answer with our experiment, whether different gravity environments would affect the performance of organisms. So the organism that we tested was Bacillus, was the same species, not the same strain necessarily, but the same species that was tested in space. So it is considered an extremophile from different points of view. We don't know exactly all the different effects that different gravity levels has on different biological models. This is an ongoing scientific effort. We know some things like the microgravity environment was shown to induce more, to increase pathogenicity of some microorganisms and also increase proliferation rate of yeasts in space. So there are some things that we already know. But we are talking about a difference between 1G level on Earth and a microgravity environment, which is basically lack of gravity. And it has this name, microgravity, because it's literally 10 to the 6th order of magnitude difference between the two G levels on Earth and on ISS. So there's still some very, very remaining gravity there, but it's micro level, it's a micro G. When you think about the moon or Mars, the difference is so small that we don't know if there is going to be any difference. Even, of course, there's going to be some difference for bone growth, and muscles between moon and Earth. But Mars, the gravity on Mars is like a third of the Earth's gravity, and the moon is 1/6. So the effects will not be so dramatic as the difference between Earth and ISS. So we have to do more experiments in these different types of gravitational environments like Moon and Mars, because most of what we know is microgravity. And we are not going to live in microgravity forever, for a long time. If we settle space, it's going to be on a planet or somewhere with some level of gravity. And that's what we need to research more, the effects of Moon and Mars on biological systems.
So one more question by Sharan. Can the radiation effects on extremophiles be studied by taking them from Earth to the Van Allen belt?
From picking them from Earth with what?
From the Earth, from transporting the extremophiles from Earth to the Van Allen belt, like the.
To the Van Allen belt. Yes, yes. I think this could be done, or we can think about experiments to do that. But we also have a lot of simulation facilities here in the lab. When we think about a space mission and space flight, we have to think about the difficulties and the technical issues that could arise in an experiment. I think we can learn a lot by simulation experiments on Earth. We can do more controlled experiments with more replicates. It's much, much cheaper to do this. We can simulate the radiation environment in the Van Allen belts, in the high radiation environment. and learn a lot. But we can also do experiments. For example, there's one mission from NASA called BioSentinel. BioSentinel NASA is sending a yeast experiment to verify the effects of galactic cosmic rays on yeasts in a satellite that will be sent in the deep space. I think it's going to be close to the moon or something. And so these experiments will verify the effects of radiation on different yeast strains. And so that's one type of experiment that is bringing a form of life to space to verify the effects of radiation on these biological systems. And we can think about variations of this experiment using more radiation-resistant organisms. But the thing is, the radiation-resistant organisms, the levels of radiation that they can withstand are orders of magnitude higher than a human cell. So there's no direct applications in terms of the level of radiation that they can withstand. We can think about like planetary protection applications, understand what are the limits of life regarding radiation, and these could drive our understanding to be able to constrain environments where we can find life. For example, places that we thought that could never host life, now we think they can. The very high environments on the moon Europa from Jupiter, it's considered a very high radiation environment, but there are some organisms on Earth that could survive there. So we have to think carefully about designing a mission to a lander on Europa because of planetary protection issues. So these, I think it can be done, but we have to think about the technical issues, the technical parameters, technical difficulties. Yeah.
Okay, so one more question. Is it that only some organisms have evolved the formation of magnesium 2+ complexes, even though it is good for survival in extreme conditions, like us humans and other organisms don't have that mechanism?
So the main hypothesis that explains the evolution of radiation resistance is the adaptation to dehydration, because there's no place on Earth that can deliver such high amount of radiation naturally. So in terms of evolution, we think that high radiation resistant organisms evolved because they are also resistant to dehydration. And humans don't have this capability because we need plenty of water to exist. So these are only organisms that evolved in environments where there were at least cycles of hydration and dehydration. So they had to withstand this type of environment. And of course, when we talk about evolution, we have to talk in terms of the ones that were selected. There's no intention in evolution, right? They were not thinking about adapting themselves. As the ones that were adapted to this environment were the ones that survived and passed their genes through time. So that's why we don't have this capability. But it doesn't mean that we don't think about strategies to use this capability somehow to benefit humans and future astronauts.
So, my question was during the primitive conditions of Earth, in the beginning of the Earth and life was still forming. During that time, the atmosphere was quite extreme, right? So they probably had those genes, to survive in those conditions, right? So why didn't those genes, why didn't we conserve those genes and so that we could unlock those whenever needed?
Yeah. So there is always a cost to maintain genetic information in our cells, because we have to replicate the molecule. We have to use the building blocks to make more molecules. So I think if you don't use a gene for a given amount of time, it will most likely lose this information. So I think basically because of that, we don't have not only that, but many other features, many other genes that are used for other environments. We don't have them in our body. Otherwise, we would have a single organism with all the genetic information available. So that's how evolution works. If you don't use a feature, if there is some information that is considered in excess, in the cell, it tends to be lost. Of course, it doesn't mean that all the information we have in our DNA is used. We also have some information in our DNA that we don't use. We don't lose all the information that we don't use. But I think it's simply a matter of how costly it is to keep an information in the genome that we don't use. We tend to lose this information, and we lose most of them, but we also have some information in our DNA that we don't know what they do. We're still learning a lot by the Human Genome Project.
So last two questions.
Mm-hmm.
The research on survival of extremophiles is to hypothesize the presence of extremophiles on other worlds, or is it to study the effects of them when taken from Earth to the other worlds?
I think I didn't understand the second half of your question.
Like, is the research to hypothesize the survival of presence of extremophiles on the worlds, or is it to study them when they are taken from Earth to exoplanets or in space?
Yeah, well, we basically can learn a lot from extremophiles, and we can certainly think about other worlds, and exoplanets with different kinds of extremophiles that we know here from Earth. So we can conduce simulation experiments. This actually has it's an ongoing topic of research. There are many different researchers doing this, and also using the knowledge of extremophiles on Earth to characterize potential biosignatures on exoplanets. This is going to be a very hot topic. It's already a hot topic, but it's going to increase a lot when we start to get the first information from exoplanet atmosphere. So we will have to distinguish what could be a biosignature. And I think the study of all life forms on Earth, and particularly extremophiles, will be key to understand what could be biosignatures on exoplanet atmospheres.
So, and one last question. You're talking about cyanobacteria producing various organic compounds on Mars, so which would be useful for plants or heterotrophs to survive on those planets. So also in the soil of those planets, we have other harmful substances also, right? So how do you plan on getting rid of those harmful substances?
Harmful substances like what's the name of this oxidant? There's different types of substances that are very harmful on Mars. We already have a good knowledge on the mineral composition and all the types of different organics that already were found on Mars, and also potential powerful oxidants. We can do simulation experiments here on Earth and select organisms that can withstand these challenging environments, and then conduct more experiments with them using synthetic biology to enhance their capability to produce a lot of different materials and compounds that will be used for growing other organisms and for producing materials to benefit humans on Mars. So basically, we can screen for organisms that can withstand the current Martian environment, and then based on that, we can select those that could survive, could grow, and enhance their capability to be useful for us.
Okay, sir. Thanks a lot, sir. It was a great talk, and thanks for the explanation.
Thank you. Thank you for the questions.
So there's a question in the chat box. I'll read it out. Why don't we send microbes to other planets, so after several billion years it'll terraform it?
So there are ideas for sending microbes to other planets and do exactly that. But there's technical difficulties and there are some ethical problems. When we think about a project as large as this, there is a lot of things that we have to think about. First, is it really going to terraform it after millions of years? What is really necessary to support these organisms, these microorganisms in other planets and be able to terraform it? Or, there is a lot of chance for them not to succeed in terraforming. We could, for example, ruin our ability to detect life if we send microorganisms to Mars, for example, and then they grow for a little bit and start to colonize a small region, and then they die, and then future missions we won't ever be able to argue that they found life on Mars because we already sent. So it's very tricky, I think before doing that, we need to make sure, in the case of Mars, that there's no life forms thriving right now there. Of course, to be able to say that with 100% certainty, we would have to look the entire surface of Mars and their subsurface, which is impossible. But we now have a very good idea that Mars is, if it has some life on Mars, it's going to be very deep and it's not very plenty. This is like only for Mars, but there are other environments on the solar system that has plenty of water and could have a lot of different life forms. But we have to think carefully about that. I think this can happen in the future, but we have to characterize and be able to control the experiment very, very, very carefully. It's not only a matter of sending things to these other planets and expect that they will terraform it, because they also can transform in an environment that is not Earth-like, so it's not going to be useful for us. So there's all these questions that we need to think when thinking about terraforming any place.
Okay, sir. Got it.
If there are any other questions, you can unmute your mic and ask, or put it up in the chat box. Okay, I think looks like there's no other questions. So once again, thank you so much for taking time out of your busy schedule to give this wonderful talk. We all really enjoyed it.
All right.
Thank you.
Thank you very much. And I hope you check out the Blue Marble Young Scientist program and the Amity University opportunity, which I think is wonderful. So
yes,
take advantage of this. Yes. Thank you very much for the opportunity.
Thank you, sir. Yeah.
Thank you.